Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive, where we crack open groundbreaking science. Today, we're looking at, one of molecular biology's really quite astonishing discoveries.
That's right.
For decades, scientists mostly thought that the cell's protein factories, the ribosomes, were like these specialized chefs. Each one just made one specific thing.
Dedicated tasks.
But what if we told you that the truth, revealed by this huge 1961 paper, was actually completely the opposite?
Yeah, totally counterintuitive.
What if ribosomes were more like, say, general-purpose 3D printers? They could make anything, just needing instructions from some kind of rapidly changing, unstable carrier.
And that carrier, that discovery, was messenger RNA. It just fundamentally rewrote our picture of life itself.
It really did.
It's such a classic aha moment in science, isn't it? This realization that a temporary, almost disposable blueprint tells the cell exactly which proteins to make.
Giving it incredible flexibility, right?
Exactly. Flexibility and really precise control. So yeah, we're going to deep dive into the, frankly, elegant experiments that prove this. It's just a beautiful piece of scientific work.
Okay, so let's set the scene a bit. Imagine you're a scientist, late 1950s. The central dogma is just coming together.
Right, the basic flow. DNA makes RNA, RNA makes protein. It was the framework.
There was this massive puzzle smack in the middle, which RNA was actually carrying the instructions from the DNA genes to the ribosomes, the protein factors.
Exactly the problem. I mean, ribosomal RNA, rRNA, it's super abundant in the cell, very stable. So lots of people thought, okay, maybe that's the carrier.
But it didn't quite fit, did it?
No, because genetic studies were already showing cells could switch protein production on or off really quickly, for specific enzymes.
Like flipping a switch.
Precisely. And that rapid response needed a quick temporary message relay, something the stable RNA, which looked more structural anyway, just couldn't provide.
So the huge question hanging in the air was, does this short-lived RNA copy of the DNA instructions even exist?
And if it does, how fast does it come and go compared to the stable stuff?
Yes, solving this wasn't just about finding another molecule. It was about linking gene control directly to making proteins in real time. Moving from ideas to like an actual molecular pathway.
The missing link.
Absolutely.
And this is where the brilliance of Brenner, Jacob, and Meselson comes in. They picked bacteriophage T2 phage infection, a really clever model system.
Why was that so good?
Well, when this virus infects bacteria, almost right away the bacterial DNA just shuts down. But protein synthesis, it keeps going.
Oh, interesting.
Yeah, but now it's making phage proteins. And here's the kicker. It does this without any net increase in stable RNA. A total paradox.
Okay, wait, so proteins are being made, phage proteins, but no new stable RNA. How's the information getting there?
That was $1 million question. So they laid out three main possibilities, three models. Model 1, they called it the classical model. The idea was phage genes make new, unstable ribosomes specifically to synthesize phage proteins. So the ribosomes themselves are specialized and temporary.
Okay, so throwaway ribosomes, basically.
Kind of. Then there was Model 2, a bit more out there. It suggested proteins were made directly on the DNA itself.
Wow, okay.
And in that model, maybe some special RNA molecule would enter the old ribosomes and just, shut them down.
Complicated. And the third?
Model 3 was their baby, the messenger hypothesis. It proposed a special, unstable RNA molecule. The messenger carries the info from the phage DNA to the existing non-specialized ribosomes.
And those existing ribosomes just read the message and make the phage protein.
Exactly. But for that to work, the messenger RNA had to be unstable, short-lived.
Okay, three very different ideas. How on earth do you test that?
With some seriously powerful techniques for the time, isotope labeling combined with density gradient centrifugation.
Ah, the heavy and light atoms trick.
Precisely. You grow things with heavy isotopes like heavy nitrogen, 15N, or carbon 13C, making their components denser. Then you can separate them from normal light components by spinning them really fast in a density gradient. heavier stuff sinks further.
And they did their homework first, setting up controls, right? That seems crucial.
Absolutely meticulous. First, they showed that heavy ribosomes grown with 15 and 13C and light ones from normal 14 and 12C medium did indeed form separate distinct bands in the gradient.
Proving the separation worked cleanly.
Exactly. It showed the technique was precise and, importantly, that heavy and light ribosomes didn't mix or form intermediate bands on their own.
Okay.
But they went even further. What if the ribosomes were just clumping weirdly during the prep?
They're like...
So they took heavy and light ribosomes, broke them down into their subunits, mixed the subunits, and let them reform ribosomes.
Ah, so if subunits mix randomly, you'd expect hybrid, intermediate density ribosomes.
That's what you'd think. But surprisingly, they still got just the distinct heavy and light bands. Maybe in different amounts, but no hybrids.
Wow. Okay, so the units weren't swapping parts during their specific prep method.
Right. It confirmed their separation method was robust and reliable for tracking whole ribosomes.
Okay, methods validated. Now, how did they actually hunt for this hypothetical new unstable RNA?
So they infected E. coli with the phage. Then they gave the cells a very short pulse of radioactive uracil, 14C uracil.
Uracil goes into RNA, so that labels any new RNA being made.
Exactly. Then quick separate the ribosomes using the density gradient.
And what did they see?
Their first key finding, this newly made radioactive RNA was mostly found with a specific fraction of the ribosomes, what they called the B band.
Okay.
And crucially, they could detach this radioactive RNA just by lowering the magnesium concentration.
Ah, so there wasn't like permanently built into the ribosome structure.
Precisely. It suggested it was something associating with the ribosome, maybe temporarily.
Okay, that fits the messenger idea. But Model 3 also predicted it had to be unstable. Did they check that?
They absolutely did. That was the next critical step after that short radioactive pulse.
The 14C uracil pulse?
Right. They added a large amount of non-radioactive uridine. This is called a chase. They let the cells grow for about 16 minutes.
So any new RNA made after that wouldn't be radioactive?
Correct. And what they saw was dramatic. The amount of radioactivity in that B-band fraction where the new RNA was dropped by about 4 times.
Whoa. So the radioactive RNA was disappearing fast.
Exactly. A rapid turnover. It proved this new RNA was metabolically unstable, made quickly, degraded quickly, just like the hypothetical messenger needed to be.
That's huge. That really supports Model 3 and starts to damage Model 1, the unstable ribosome idea.
Oh, it does. But they needed to kill Model I completely.
How did they do that?
With, I think, the most elegant experiment in the paper. They grew bacteria in heavy isotopes, 15N and 13Cs. All the old ribosomes were heavy.
Okay, heavy bacteria, heavy ribosomes.
Then they infected these with phage and immediately switched the bacteria to a light medium normal 14N12C.
Right.
And at the same time, they added radioactive phosphate 32P, which labels new RNA.
Okay, so if Model I is right, new unstable ribosomes should be made after the switch. They'd be made from the light materials.
And they'd contain the radioactive 32P label, so you'd expect to see radioactive light ribosome.
Makes sense. What did they actually find?
This is the clincher. The new radioactive 32P labeled RNA wasn't in the light region at all. It was found associated with the density of the original heavy ribosomes.
No way. So the new RNA was latching onto the old heavy ribosome?
Exactly. No new light radioactive ribosomes were being made, period.
Wow. Okay, so Model 1 is out, dead. Ribosomes aren't being turned over.
Gone, which left Model 2 synthesis on DNA versus Model 3, the messenger on old ribosomes.
Right. So how do you tell where the proteins are actually being made?
You track the proteins themselves, similar setup. Grow cells in heavy medium, 15N. Infect with phage, switch to light medium, 14N. But this time, pulse with radioactive sulfur, 35S, which specifically labels newly made proteins.
Okay, follow the radioactive proteins. Where do they end up?
Bam! The results were crystal clear. The 35S label, the new protein, was found only in the band corresponding to the pre-existing heavy ribosomes.
Only the heavy band.
Only the heavy band. Absolutely no radioactive protein label showed up in the lighter region where any hypothetical new ribosomes would be.
So protein synthesis is definitely happening on the old ribosome.
Unmistakably. It wasn't happening directly on the DNA, ruling out Model 2.
Okay, but could that 35S just be some stable ribosomal protein being labeled slowly?
Good question. They checked that too. They did another chase experiment, this time chasing the 35S protein label with non-radioactive sulfate.
And
they found almost all the 35S radioactivity could be washed out chased away.
Meaning it was intransient, newly made proteins, not stable structural parts.
Precisely. It confirmed again. Protein synthesis happens on the old pre-existing ribosomes, using instructions delivered somehow. Model 2 was out.
Leaving only.
Model 3, the messenger hypothesis.
So let's recap the evidence, because it's pretty overwhelming when you put it all together.
It really is. First, phage infection, no new ribosomes are made. We saw that clearly.
Well checked.
Second, a new unstable RNA appears. It turns over rapidly, matches the phage DNA composition, and temporarily sticks to the old ribosome.
Check. Got that too.
And third, protein synthesis. Pretty much all of it happens on those old ribosomes.
Check. And those points only fit Model 3, right?
Perfectly. The only explanation left standing was this unstable messenger RNA carrying the genetic code from DNA to the ribosome workbench.
And they found hints this wasn't just a phage thing.
Yeah, that was also crucial. They observed similar unstable RNA behavior even in uninfected cells, suggesting this wasn't some weird viral trick, but a fundamental process of life.
Wow.
They even characterized this mRNA a bit, found it was a minor fraction, variable in size, and crucially, its building blocks, its nucleotide composition, mirrored the DNA's composition.
It's just such a complete picture they built. This wasn't just another finding, was it? It feels bigger.
Oh, absolutely. It was a genuine paradigm shift. The field desperately needed a clear answer on how gene information gets used, and this paper just delivered it. Bang!
It filled that gap in the central dogma.
Completely. Identified the physical molecule responsible for carrying the gene's instructions for protein synthesis.
What was so innovative about how they did it?
I think it was the combination, the elegant conceptual design, thinking through the models, the predictions paired with really powerful physical methods.
The isotopes and the centrifuge.
Right. That combination gave them both time resolution, seeing the rapid turnover with the pulse chase and physical separation, isolating the messenger RNA associated with ribosomes away from other RNAs. That let them pick out this unstable message from the stable background noise.
And there's another group publishing similar stuff at the same time.
Yes. In the very same issue of Nature, Gross and colleagues published complementary findings that really helped solidify the whole concept, and the name messenger RNA quickly stuck.
And the collaboration behind this paper, Brenner, Jacob, Meselson, that was pretty special too, wasn't it?
Incredible. A tricontinental effort. You had Jacob in Paris, the master of bacterial genetics, Brenner in Cambridge with his molecular vision and experimental boldness, and Meselson at Caltech, the expert in physical chemistry and ultracentrifugation.
Wow. Different skills coming together.
Exactly. That synergy, plus, the intense scientific competition of the era, really pushed this forward. Their combined genius theory, imagination, rigorous method created an experiment that just beautifully read out the flow of information.
It turned RNA in the central dogma from just a step on a diagram into a real working mechanism.
Precisely. It established ribosomes as these stable, general-purpose decoding machines, just waiting to be programmed by transient messenger RNAs. And that principle, it underlies basically everything we know about gene regulation today.
Paving the way for, well, mapping genes, understanding how cells work, right up to mRNA vaccines today.
Absolutely. It's foundational.
It's amazing. But, even after all that, after showing the information isn't in the ribosomal RNA, it does leave a lingering question, doesn't it?
It really does.
What is the ribosomal RNA doing then? If it's not the message carrier, what's its precise job?
Exactly. If the ribosome is just a non-specialized machine reading the tape, what's the role of its own very abundant, stable RNA component in that whole complicated dance of making a protein?
Something to think about.
Definitely a thought to mull over long after this deep dive.